Good morning, students. This is Nikhil Kumar, UGT Biology from JNU Dakshina Kannada, with you to discuss the chapter Cell, the Unit of Life. This topic is not new to you because we are already discussed this topic in the junior classes. We well versed about the cell, but here we are going to study more detail about the cell and as well as its components. So what you are seeing over here, yes, a brick wall, and the wall is made of course, it is made of bricks. So in that sense, we can call the brick is a unit of wall or unit of a house. But when you come to the organisms, whether it is microscopic unicellular organisms or multicellular organisms like this mushrooms, fungi, or jellyfishes, or a plant, or even higher animals, and the most complicated animal, what we say, all these organisms are made up of cell or cells. That is why the cell is called as the unit of life. Now let us explore the cells and the structure of the cell, components of the cells and its functions. Ready? You may heard about this gentleman named Robert Hooke. Yes, he's a man. First seen a cell under microscope. We cannot feel to call it as a microscope. Even though his first invention, in just remember, in 1665 he has observed that, and he has seen, like you see that this is what he has seen and the pictorial representation. So this is his instrument, so called the Hooke microscope. Got it? And he called them as cell. These stories and all we know. Now later on, let us see little history about the cell. So from the Robert Hooke, the next scientist who take over the activities in the cell towards the cell theory discovery is Antoine van Leeuwenhoek. What is his contribution? He is observed the living microscopic organisms. Living organism is first discovered by Leeuwenhoek, and his structure or his microscope was so rudimentary, what we can all see here by using a lens, etc. And this is what he has recorded, drawn diagrams of the living organisms that he has seen in water sample, and he called them as animalcules. Interesting. Later on, Robert Brown, another scientist, so it is around 100 years later, more than that, 1831, his discovery, he discovered the nucleus. Now he has seen nucleus like this, and now when we are doing in our laboratory, we see nucleus like that, because we had done this onion peel experiments. In that we have seen nucleus like that, or sometimes like this, depending on the staining. So this is what the nucleus, the prominent structure, and the cytoplasm and the cell wall. These are the things what we can see in our laboratory with our normal compound microscope. Right? So later on, as an invent of electron microscope, the most sophisticated instrument, in the cytology or study about cell, we could able to see the detailed structure of cells. So the electron microscope, which is using a flow of electrons, we know the structural and as well as the functional capacity of this instrument. We are seeing a lot of science could able to explore lot of things. They have seen this in cell organelles inside the cell like Golgi apparatus, mitochondria, chloroplast, and endoplasmic reticulum. Later on, what happens? Colored electron microscopy came. With that, we could able to see the color of the may not be the real color or fluorescently colored cell or cell organelles we could able to see like this RBC and as well as this bacteria by using electron microscope. Let it be there. Before that, a group of scientists has contributed for the formation of cell theory. Cell theory is very simple. Of course, this all begin from Robert Hooke, but two other scientists, Schleiden and Schwann, that is Matthias Schleiden and Theodor Schwann, the two scientists, they had told or they had discovered that all living organisms are made up of cell and their product. Either they are unicellular or multicellular. They all made of cell and the product derived from the cells. That is the contribution of Schleiden and Schwann. So this is the core idea of cell theory. But they could not say from where these cells came. So with that concept, another scientist came. His name is Rudolf Virchow. Rudolf Virchow has told that all cells will arise from pre-existing cells. It cannot came from non-animated matter. All the cells what you are seeing is coming from a pre-existing cell. From a parent cell only a daughter cell will form. Like that Rudolf Virchow has told. So with that the cell theory is now at present condition. What is cell theory says? It mainly says two basic statements. The first statement that is given by Schleiden and Schwann. All living organisms made up of cell and their product. Later, Virchow's contribution. All cells arise from pre-existing cells. So this is our cell theory. Okay. Now we are going to the explore the cells. So this is what we are seeing. Already we told about onion peel cell under microscopes and human cheek cells. In both the cells, what we could able to see only three things. In the animal cell or human cell, that is the plasma membrane or cell membrane. B is a nucleus and C is a cytoplasm. And here also we could able to see the cell wall, nucleus, cytoplasm. Sometimes we can see the vacuoles also. And when it comes to the condition of prokaryote or eukaryotic division also, what we will understand that is almost all the cells having the same machinery, and whether it is a plant or animal or prokaryotic cell, the main area of cytoplasmic activity or cellular activity is in cytoplasm or cytosol. In certain higher classes we will study as cytosol. Here itself we can study as cytosol or cytoplasm. So whether it is an animal cell or plant cell, and this all takes place in the cytoplasm, which is like the protein synthesis, right? The production of other cell organelles, expression of the genetic material. So this all basically takes place in the cytoplasm, or cell organelles in the cytoplasm, energy production, photosynthesis. So these are all. Okay. So whether it is a largest smallest cell, I told you already. This is the largest cell that we know at present, the ostrich egg, to the smallest cells in human body. I'm not telling this is not the smallest cell in the whole living system. That is PPLO. We are going to see that. The human body, that is a sperm. Whether it is a sperm or the largest ostrich egg, the machinery is almost the same. Like nucleus, what are cell organelles are there, Golgi apparatus, mitochondria, vacuoles. This all machinery is almost same. So that is what we are going to see one by one. First, we are going to see the prokaryotic cell. This is what I told you, the smallest organism or the smallest living cell, that is PPLO, Pleuropneumonia life organisms. So they are the variant of mycoplasma also. So there will be different type of organisms. There will be bacteria. It is not as big as we are seeing here. Okay. And blue green algae. We know that blue green algae, small cyanobacteria, mycoplasma, whatever it is, they are comes under the prokaryotic. Prokaryotic. So why do they call this prokaryotic? We will discuss. Okay. When discuss about bacteria, we have studied in the first unit also. Bacteria may be of different type. That is bacillus type is here. That is a rod shaped. As you are seeing over here, the rod shaped bacteria is here. Coccus, Streptococcus pneumoniae, pneumonia causing bacteria. And Vibrio cholerae. So spirillum or spiral shaped bacteria is here. So different Rhodospirillum. So Bacillus thuringiensis. So different type of bacteria are there, and still there are a lot of modification is there. Actually, coccus also, cocci, if it is more in number or 2 in number, there are different names. So we are not going to detail about all these things. Basic difference of 4 type of bacteria. Okay, this is the basic structure of bacteria also. Right. Now let us see the layers of bacteria. Basically, bacteria having 3 layers. We have seen in the previous slide also. The outer one, it is a glycocalyx. It may be a capsule or a slime layer. Okay, so you can see it is maybe a capsule or slime layer. And the second one is cell wall, and the third one is plasma membrane. And the internal structures we can see here. Flagellum we can see that. We can see the plasmid DNA, we can see that. Ribosomes we can see that. PD-like structures. Okay. The nucleoid region, the genetic material you can see over here. Okay. So what is a glycocalyx? It's okay. The glycocalyx, if it is a loose sheet-like structures in some bacteria, it forms a slime layer, slimy, slippery layer they forms over the bacteria in certain bacteria, as you are seeing over here. Slimy, slippery layers they form. In certain bacteria, it forms, the glycocalyx forms a tough capsule, protective layer capsule you can see. Okay, so it may be different. That is a variant of glycocalyx. It may be a loose sheath, so slimy layer. As you see the plaque formation in our teeth, that is a slimy and slippery, formed by the slime layer containing bacteria, isn't it? So tough capsular bacteria is also there that will protect them from acid or some attack from outside environment. They can be saved. So there are different ways the glycocalyx help the bacteria. Okay, so next let us see about the genetic material of bacteria. So, and we know that is a prokaryote. We had studied about that. Pro, it's a primitive one. Genetic material is a primitive condition. That means a nucleus or double membrane cell organelles are not there in prokaryote. So that is why the genetic material is not confirmed inside the double membrane structure nucleus. So we call that aggregation of genetic material as nucleoid. So that is a main DNA of bacteria. You can see over here nucleoid is labeled here. So this is the nucleoid structure of bacteria, the prokaryote. Then what else? Bacteria is having a plasmid. Okay, this is the new information for us. A plasmid. What is a plasmid? So plasmid is an extra chromosomal circular DNA. So where is the main chromosomes in bacteria? The main chromosome or the main DNA we call as nucleoid is here. It is beyond that, another type of circular DNA that is present in the bacteria, that is called as plasmid. So you can see the plasmid over here. Plasmid have a lot of role. Main one is certain plasmids give resistance to antibiotic. You may heard about that is antibiotic resistant bacteria. That resistance they are getting from the plasmid. The plasmid carries that gene. We got that importance. And plasmid is also useful in bacterial transformation. So they can transfer this plasmid from one copy of this plasmid from one bacteria to another bacteria. Thereby both can be disease resistant. Their number can be increased. The another structures are mesosome and chromatophores. So these are extension of plasma membrane into the cytoplasm. What we can see over here, that is the mesosome we can see here. Now it is projecting inside, right? So that is an extension into the plasma membrane. So they have great role producing inclusion bodies, producing the cellular machineries, etc. Chromatophores. Actually, that is a photosynthetic bacteria that is present, that is containing chlorophyll-like structures, bacterial chlorophyll, etc. Right? So the role of mesosome: cell wall formation, DNA replication, and distribution. So they do these functions to the extensions into the plasma membrane like mesosome or chromatophores. Great role in cell wall formation and DNA replication. Got it? So this is about the genetic material and as well as inclusion or extensions of plasma membrane into the cytoplasm of bacteria or prokaryotic organism. General for prokaryotes. Now another thing we are going to discuss in common in prokaryotes are the flagellum, the locomotory organs. There are basically three parts of flagellum. That also we are going to discuss in the end of this chapter about the structure of filament and the hook and the basal body. Got it? So this you can see here the filament part of the flagellum, right? A bacteria you can see with the flagellum, or a microgram native bacteria is a flagellum you can see here. With the filament is here, the basal body, the area where it is attached with the cell wall and components you can see here, and here there is the hook region, and the basal body we can see over here. Okay, so this is what we are going to study in detail in the almost end of this chapter. Then when we come to the other organelles or other extensions of the microbes, that are pili and fimbriae, it may not be necessary that these all components like flagellum, pili and fimbriae present in the same organisms. Some organisms may contain pili and fimbriae. Some organisms may contain flagellum and pili. So it all varies. Got it? But here it for a typical structure as shown to depict all the layers, all the type of locomotory structures. So what may be the function of flagellum? Maybe that is helping for movement, okay, locomotion in the media. By flapping the flagella, they may move. Okay, and even we know about cilia of some organisms like parameciums and that. That is not coming into the picture here. Here they having the prokaryote having here the pili and fimbriae. You can see the pili and fimbriae almost similar structures, but pili are the longest one and the fimbriae are the small one. Compared to these two, I am not telling compared to flagellum. Pili is longer and fimbriae is smaller. So how does it look like? So it is not having a role in mobility. Flagellum having a role in mobility. What is their role in attachment? Maybe see in the strap of watch strap or the chapel strap. We can bind to these two together and remove the strap. Okay, how do they attach this? Just go through that. You can see the small thread-like structures and hook-like structures in both the surfaces. Similar type of things the bacteria uses here, the fimbriae, to attach on the surfaces, whether it is a rock or the host cells. Okay, so role of this extension, cellular extensions, 3 times. Okay. Now let us see about the coloring of bacteria or staining of bacteria. So basing on the staining procedure, we call it a gram staining. Okay, gram staining. So we call the bacteria as gram positive bacteria and gram negative bacteria. Gram positive and gram negative bacteria. You can see the gram negative bacteria and gram positive bacteria. So why do they call us gram negative and gram positive bacteria? Very simple basic things we will study here. We are not going to the detail about the cell wall composition or the complexity of the bacteria which help them to stain differently. Got it? So you can see that another double staining procedure. Actually, gram staining means a type of double staining procedure. First, stain the bacteria, given bacteria with crystal violet. Okay. Then decolorization and counter stain with safranine. Safranine is a stain available in our laboratory. Sometimes crystal violet also may be available. So these two that together, that is called as what? Gram staining. And after this procedure, if that bacteria is this purple color, violet purple color, then we can call as that a gram positive, and if it is reddish in color, then we call as gram negative. Okay, if they catch the gram stain, crystal violet, then gram positive, not, then it is gram negative. That is actually happening because of a small complexity. You can see that here. See that the cell structures, again, not required actually, that peptidoglycan, the outer structures, okay, both amino acids and as well as carbohydrate component, that only actually helping for this catching the gram stain. So that is gram staining. But here when it gram negative bacteria, they have another structures, okay, outer that. So that is a, they will not catch the gram stain. So this is what the basic thing you should be able to understand about gram staining. Okay. So now let us go into the cell organelles of the bacteria, little detail. That is first we'll study about ribosome. So you may heard about ribosomes, right? They're very small, 15 to 20 nanometers. Nanometers, we know 10 raised to minus 10 meters, and they have two subunits. A ribosome is two subunits, 50 years and 30 years. What is this? 50 years and 30 years. S is Sedberg unit. Actually, we can call it a sedimentation coefficient. Here you can see in this bacteria, we can see a lot of ribosomes. Got it? So these ribosomes are here mentioned. Okay, So this is our two subunit, 50 years and 30 years. Together it is 70 years. This is not mathematics. This is biology. 50 plus 30 is, sir, it is 80, sir. No, but here it is when this both came together, the sedimentation of that, sedimentation, during centrifugation and all, sedimentation coefficient, the rate of sedimentation of items, denser item will go bottom and the lighter one will come up. So this is the ultra centrifuge sedimentation coefficient of ribosome. It is 70 years for the prokaryote while 80 years for the eukaryotes. So this is 50 years and 30 years. Let's start studying from here. Again we are going to study in detail about ribosomes. Okay. So these are two subunits we can see, 30 and 50 years. 30 is a small subunit and 50 is a bigger subunit. Got it? Okay. And another one is they are helping in protein synthesis. And they may form a structure called as polysomes or polyribosomes. So what is this polysome, polyribosome? Immediately after saying about ribosome, we are telling all these stories. Of course, ribosome we may saying once again, but just see here what is the story of protein synthesis and all. We have to tell in detail actually, that we are going to study in detail in class 12th also. So protein synthesis all begins from DNA, as you are seeing here, and DNA make the copies as RNA. So the RNA which is required for protein synthesis or using for protein synthesis we call as mRNA, messenger RNA. Just I'm telling the basics. And on that RNA, ribosomes will attach for what purpose? Protein synthesis. Ribosome will go through the RNA, thereby they will form the poly amino acids and proteins. Got it? So if numerous ribosomes attached at a time in a mRNA, it is called as polysome or polyribosome. When human being case or higher animals case, eukaryotic case, there will be only one ribosome on one mRNA at a time. So the polysomes or polyribosome is a basic feature or exclusive feature of prokaryotic organisms, prokaryotic protein synthesis. Okay, so here only we can see DNA, mRNA, proteins all together. That is different in human being case, while DNA found only in nucleus, while the RNA and as well as protein synthesis can take place in cytoplasm. As there is no nucleus in bacteria, all these things takes place in their cytoplasm. Okay, so ribosome and the rolled structure we understood. Okay, now next is inclusion bodies, that is specifically found in these prokaryotes. That is storage bodies, like just like we can see here the inclusion bodies here. Small vacuole like structures, inclusion bodies we can see. Maybe phosphate granules, cyanophasian or glycogen granules, gas vacuoles, and different type of inclusion bodies may be there. Right? So you can see the diagram of a prokaryote once again, the detailed structure of that. Now let us see about the eukaryotic cells. What makes it different from prokaryotes? We know eukaryote. So they have a nucleus, nuclear envelope, and special structures for locomotion or cytoskeleton. What is cytoskeleton? Skeletal framework inside the cytoplasm. We are going to study about the endoplasmic reticulum, Golgi apparatus, especially endoplasmic reticulum acts as a cytoskeleton or structural framework. Locomotory organism. We had studied about the cytoplasmic streaming that goes the locomotion in amoeba. So this all comes under eukaryote, including human being, higher plants, etc. So we can see the basic types of eukaryotic organisms. Unicellular organisms are there, like fungi and protista. Can you recall one unicellular fungi that is shown here? That is a yeast. Yeast is one of the unicellular fungi, not all fungi. Okay, Protista. Already we have studied about Protista. That is amoeba, isn't it? Paramecium. So these are amoeboid protozoans. Then multicellular organisms are there, like plants and animals. These are all eukaryotic organisms. And we see that eukaryotic cell is more richer than that of prokaryotic cells. You can see the central big nucleus. This is animal cell, of course, and around that we can see that endoplasmic reticulum. So that makes the cellular cytoskeleton or network. Then you can see the Golgi apparatus, mitochondria, and centrioles. What is secretions, vesicles we can see here. A lot of other components we can see over here, that is vacuoles, vesicles, lysosomes. So here I have seen the cytosol. I told you cytosol, the word used for cytoplasm. So villi extensions are there. So this is an atypical eukaryotic animal cell. So when you come to that, you can see again animal cell structure in detail. So just see the specific features of an animal cell. Okay, you can draw from a textbook also. It's easy to draw, but outer boxes just avoid. So you can see here the basic structures. It start from the plasma membrane. Inside that, what is shown, the cytoskeleton elements. Framework will be there, proteinaceous. Then mitochondria. Mitochondrion is the different terms using. Then you can see the lysosomes. Then Golgi apparatus. Endoplasmic reticulum. Peroxisomes. Ribosomes, very small. Rough and smooth reticulums you can see. Again the nucleus, inside that nucleolus, genetic material, chromatin, envelope with the holes, nucleus, centrioles are there. So these all the basic structure. You can list out, please list out the specific features, or what are the features what you are saying in this animal cell. Then similarly we'll see the plant cell, the specific exclusive feature of a plant cell. Similarly, the nucleus is almost the same, and endoplasmic reticulum both are there, smooth and rough. Golgi apparatus is there. Another important feature what you are seeing is a big vacuoles. Peroxisome is there, mitochondria is there, plasma membrane is there. Another extra feature is that the cell wall and the chloroplast. The two things exclusively seen only in plant cell. Okay, so that's all about this session. Let us meet in another with another session. Thank you.